Hi, hello everyone, and welcome to STEM Thing to Talk About. And today on STEM Thing to Talk About, we've got Charlene Konkankit. Did I say it right? I said it wrong again.
Konkankit.
Konkankit. I'm sorry, Charlene.
That's okay.
Yeah, but so Charlene, thanks again for coming on to STEM Thing to Talk About. We're all really excited to hear from you. I guess the first thing that I'd like to ask you is just, can you introduce yourself and tell us a little bit about what it is that you do?
Sure. So I'm Charlene Konkankit. I'm a graduate student at Cornell University. I study chemistry. More specifically, I'm in bioinorganic medicinal chemistry. So basically, I try to make new molecules for treating cancer.
Okay, very cool. So new molecules for treating cancer. And one of the questions that we also had come up, Charlene, was what specific types of cancer do you treat? And in doing so, what methods is it that you use?
So we look at a variety of cancers in our lab. The one that I've focused on the most in the past 5 years, I'd say, is ovarian cancer. We also look at cervical cancer. We have some breast cancer cell lines. We also have prostate cancer and lung, which is a huge one that we're all trying to tackle these days. And then I think you asked what methods we use.
Yeah.
So it always, for us in our group, it starts at the very basic things. So at a molecular level, which atoms and where are these atoms on a structure going to be useful for treating cancer? So we always try to make subtle changes or even big changes to see how it affects their ability to kill cancer cells and their ability to only kill cancer cells versus healthy cells. So we start there, and then we have to make sure that we have the right structure, and then we want to understand how it's probably going to work inside the cell. So we do a bunch of biological experiments to see what signaling responses or what sort of cell death pathways we're starting to induce when we treat the cancer cells with these molecules.
And could you just specify what is exactly like a signaling response?
So it's more, I guess, so when you, let's say, you perturb a system, or you have, let's say, you're just resting there and then someone flicks you on the ear, and then that is like a signal for you to wake up or do something or freak out or something like that. So it's kind of like, what responses do the cells have when they're getting treated with these molecules?
That's a really cool example. I like that. Now, and I noticed that you said that you guys mostly do ovarian cancer. Is there a reason that you guys mostly do ovarian cancer?
So ovarian cancer, we've seen a lot of patients who they get treated with current chemotherapeutic agents, but then after some time, after a few cycles of getting that treatment, they experience the relapse forms of the disease. In other words, they gain drug tolerance. So those drugs can't work anymore to treat those patients, and it's very common in ovarian cancer. So that's one reason why we're trying to study them, so that we can make alternative compounds, alternative molecules that can treat these forms of cancer without having to experience the drug-resistant forms.
Very cool. Nice. Now, another question that we had was more specifically with cancer treatments, and I think that you kind of mentioned like with radiation. One of the questions that we're having is, how does radiation exactly affect cancer cells? And so yeah, if you could go off of that a little bit.
So when you ask about radiation, do you mean radiation that induces cancer cells, or radiation therapy? So we have both. So there's, for instance, if you're exposed to a lot of radioactive sources, and for instance, the radium girls.
Right.
That's an old time story, right?
Yeah.
These women were working in a workshop, and they were painting these watches, and they would lick the paintbrush, and it had a radioactive source on them. So in that case, these radioactive sources are sending radioactive particles or rays into the system, as in your body, and it affects your DNA, you end up mutating, and then you can form cancerous cells, and then they multiply from there. But alternatively, in treatment, so there are three major forms of cancer treatment. There's chemotherapy, which is what I'm trying to work on. We have surgical resection, so when the doctor goes in and just takes out the malignant tumor. And then we have radiotherapy, so we can do things like targeted alpha therapy, or that's something we're still developing. We're always trying to develop more treatment plans. So we can use radiation in that sense to bombard a tumor and try to kill that specifically.
Okay, cool. No.
So it can be, it's on both sides.
And one of the things with radiation treatment for cancer cells is that a lot of times it ends up killing healthy cells. Is that correct?
Yeah, so that's an issue with both chemotherapy and radiotherapy. So with the radioactive sources, yeah, it can also get to the healthy cells, and you'll experience tissue damage there. That also happens with chemotherapy. So for instance, one of the major chemotherapeutic agents is called cisplatin, and that's just one of them, but a lot of them will cause like hair loss, hearing loss, or even kidney damage. So it's not just radiotherapy that will do that.
And the lab that you work in, they've done a lot of work with cancer drugs that are similar to cisplatin, correct? But you use different agents. Could you explain that a little bit more?
Yeah, so cisplatin is a platinum-based compound. That's a third-row transition metal. And we're working on, like our lab, or me specifically, I work on developing structures from the same row, but it's a different element. So I'm working with rhenium. So that's RHENIUM. So it's similar to platinum because it's in the same transition metal row. So it's fairly stable, and we're trying to figure out ways that it will kill cancer cells but not experience the same drug resistance as platinum-based drugs.
Okay, so the cisplatin drug has been it's effective, but also like there's been some resistance against it. Is that kind of?
Yeah, in certain forms of cancer.
Gotcha.
Cisplatin was really good for testicular cancer.
Interesting. Very cool. Another question that I thought that I received from my students that I thought was pretty intuitive was, what aspects of cancer cells are you trying to impede? Is it the cell structure? Is it the function? What exactly is it that you guys are working to impede?
So structure and function kind of go hand in hand for cells. You need the structure in order for it to behave properly and to replicate. So the answer is both. We're trying to do all of it, and then kind of like the responses that the cells have tend to be starts on a molecular level. So for instance, with the chemotherapeutic agent, we'll treat a cancer cell, it goes in, and then it targets one specific thing. But we don't always know what it is, and small changes to our structures can change the biological target. So for instance, I've made a compound that when you put it in, it makes the cell membrane just pop. And that's how it kills the cells.
Yeah.
That's called necrosis. And then one of my lab mates, Sierra and Peyton actually, they made a rhenium compound that's a little different from mine, but it does not cause popping up the cell membrane. It goes in and causes stress in the endoplasmic reticulum. And it kind of bombards from there. So
really you guys work with both, like impeding both, like both. That's cool. Very nice. That's good to know. Cool. Well, one other thing that obviously your research that you guys are doing is awesome and it's really interesting. But we're wondering if you could possibly kind of explain a little bit as what does your day-to-day life look like as a researcher in this field, And like, what are some of the tools that you use, and what are some of the things that makes you enjoy your job?
Okay, so now my everyday is at home.
Yeah, much like all of us. I hear that.
Yeah, mostly at home on the computer. I'm finishing up grad school, so I'm mostly writing these days. But when I was in the lab, well, it varies where I am in my project. So just to work from the ground up, let's say if I'm doing everything, maybe I have some cell experiments and maybe I need to do some chemical synthesis, so I need to make some molecules. So I start by warming things up if I need it. So if I'm going to treat cells, I need to make sure everything's warmed at 37 degrees Celsius.
And why 37 degrees Celsius?
37 Celsius is body temp.
Yep.
So that's 98.6
Makes sense.
Fahrenheit. So maybe I'll set up a reaction. So when I say set up a reaction, that means I'm trying to make something. Okay, so I'll warm up a bath, because a lot of the times when you want to do a reaction, you have to use high temperatures, or higher temperatures at least. Or if I'm going to work with cells, then I warm up growth media, which is basically vitamin or sugar water that cells need to grow. And I can treat them with that. Working on the hood is really interesting because we have two different hoods. We have a synthetic hood and we have a biological hood. So the synthetic hood, we're just being really careful with the chemicals that we're using, because a lot of them are corrosive or irritants. Some of them are carcinogens, which is ironic. But then in the biological hood, we have to make sure that we're being very careful and sterilizing everything as we go in. So when we want to work in the biological hood, we have to put on gloves and spray with 70% ethanol. So that's really relevant for today's world, because we're trying to sterilize everything these days, right? 70% ethanol is excellent. It kills enough of the bacteria that we can work in a sterile environment. There are other ways to sterilize things, especially like when we were putting things into our hood, we have something called an autoclave. So that's kind of like a steamer oven. It gets really hot and high pressure, and it just kills everything that's in there. So it's all sterile. So then when we go into the hood and we're all sterilized, then I can start working with my cells. I can treat my cells with the compounds that I make, and just see how many of the cancer cells die, so I can find out how potent my compounds are. I can also do studies to figure out how they're killing the cancer cells by taking the solution or the mixture after I'm done treating it, and just to see what proteins are being expressed afterwards. There's a lot of experiments that we do. It's very ground up in our lab. I've also I haven't worked with mice, but I've worked with post mice, if that makes sense. So I'll send my compound to people who do work with mice, and they will treat mice. And then afterwards, the mice are sacrificed. Because,
happens. The world of science.
Yes. And then they'll give me parts of the mouse, which I then analyze to see if it was toxic in certain areas of the body of the mouse. I was actually able to find that one of the rhenium compounds I'm working with does not have any off-site tissue damage. So that's very different from current radiotherapy and current chemotherapy, because we just talked about like hair loss, hearing loss for a lot of current cancer patients. But the molecule that I made, that I was working with at least, does not seem to induce any of those bad side effects.
No, and that just kind of makes me think, like, so what are like the next steps, like with a drug like that, when you see like after testing it on mice, then it's not having harmful effects on other parts of the body? Like, what is kind of the next steps there?
So we are trying to develop other molecules, similar molecules, that are more potent to cancer cells and more descriptive in terms of what we can figure out that they're doing inside. So what is the biological target? So let's say we already had a molecule that has very promising biological and chemical properties, then we can patent it, and then maybe it can be taken up by someone to go into some preclinical trials. But so far, that has not happened for us in particular. So we're just trying to develop other molecules that could be very potent.
So, like, how long would it take, like, after you, like, theoretically, like, if there was a company that decided to take up your guys' drug that you guys made, like, how long would that take before it would actually be able to be treated on cancer patients?
I don't know
Yeah.
exactly, but it's years.
Yeah.
I feel like I've seen something previously where it was like a decade,
yeah,
or even longer. So it's a long time.
it is.
Because we have to go through all of these testing parts, like
Right.
the procedure. You have to go through these to make sure that's okay, and then you do it here, and then you need to make sure that group 1, 10 years down the line, doesn't have these adverse side effects long term.
Mm-hmm.
So these are things that we have to know.
You gotta be super thorough with it.
Yeah, so that's why developing a vaccine right now is so hard too, because you also have to go through all of these.
I know, it's super applicable right now. I just have two main more questions for you, and because I don't want to take too much of your day, but one question is, so I had the opportunity, fortunately, to actually work in the lab with Charlene. And one of the things that I noticed when I was working with her was her notebook, her researching notebook. Can you just explain briefly, like, how important that is in the researching world, to keep meticulous notes in your notebook and whatnot?
It's super, super duper important to make sure you write everything down, and to be very descriptive and clear in there, because let's say, okay, and this happens all the time to me, to fellow grad students, people 10 years ago, people 10 years from now will be having the same problem where they forgot to write something down. And then they're like, oh no, what concentration did I use? I'm trying to publish this in a manuscript and I can't even figure out which concentration I used. So you have to go and backtrack. Like, maybe I wrote it down somewhere else. Maybe it's on here. Maybe I wrote the procedure and I didn't write the actual concentration post procedure. So now I have to look at my math and redo it. And it eats up all your time later, or you didn't write any of that down and you have to redo it. So it's not good to not write things down.
So yeah, hopefully some of my students out there hear that when I bug you to take notes on different things, then there's a reason. And then lastly, Charlene, if you could just let us know what kind of brought you to this world of research in science and in the field that you're in.
So I've always been interested in science. My dad, so my family has different interests. My dad is a car mechanic, and my sister is an MD doctor, and my other sister is a speech pathologist. So we all went in different areas.
Yeah.
But it all started with my dad just being very logical in his thinking, and we were watching that, and then we just kind of clicked, and then we went through school. All three of us went to the same school. We all liked different subjects. I liked chemistry the most because it seemed the most straightforward at first.
Yeah.
Balancing equations was fairly straightforward to me. Memorizing things was straightforward to me, only to go to college and then grad school to find out that the rules that I knew don't really apply anymore because I know too much.
Yeah.
So I went into college and I wanted to be a pharmacist because I didn't know what else to do with chemistry.
Yeah.
And then I realized I could do a lot of research with chemistry. So I first did some physical chemistry and material science. So I made nanoparticles and tried to use them for biosensing. And then I went to grad school and I started in solar energy research.
Okay.
That didn't work out so much for me because I wasn't as interested in solar energy as I was in the medicinal research area. So I talked to my current boss, my advisor, Justin Wilson, and I asked him if he had space in his lab, and he gave me a shot, and I mean, I'm still here, so I guess.
So it's worked out okay then. Very cool. That's awesome. Well, Charlene, stay on because I'm going to talk to you after I'm done recording. But thanks again for coming on to the show today. Really appreciate it. Yeah, of course. Thanks, Charlene.